How to go to slideshow? Slideshow button. Thank you. Second.
Yeah. Good afternoon, sir.
Good afternoon. Oh, Harsha is here.
Yes, sir.
Okay.
How are you, sir?
Oh, I'm good. I'm good. Thank you.
Thank you, sir.
Good afternoon, sir. Niranjan.
Oh, good afternoon. Niranjan, how are you?
Oh, fine, sir.
Where are you now? In Nanjali or in Chumaka?
Sir, in the department campus only.
In the campus only. Very good. Very good.
Okay.
Good afternoon, all of you. I can understand this hour of 2 p.m. listening the lecture for for all teacher participants. I can understand the difficulty of listening the lecture at this 2 p.m. in the afternoon hour, that after the lunch. Okay, because I was also a participant long back, and I can understand the difficulty of listening the lectures continuously, every day 4 lectures continuously. But obviously there is no other alternative go. We need to understand, we need to complete some of the official formalities to get our promotions done and other things. Okay. In another 1 hour or so, I will be telling you about the research activities of my group in the Department of Biotechnology, where Professor Dr. Niranjan is also the author for one of our papers. Since last 10 years or so, my group is extremely working on vibrant research on development of molecular diagnostics for the specific detection of mostly bacterial pathogens. And before going into subject proper, my dear participants, I would like to introduce this subject. As we all of us already know that global population is exploding in the geometric progression, whereas food production is increasing in the arithmetic progression. Am I audible, all of you?
Yes, sir. Yes, sir.
Yes, sir. Yes, sir.
Audible, sir.
Okay. Okay. I was telling you the introduction of my talk. The global population is exploding in the geometric progression, whereas global food production is increasing only in the arithmetic progression. Geometric progression is multiples 1, 2, 4, 16, 64 like that, whereas arithmetic progression is just continuous number 1, 2, 3, 4, 5, something like that. So we need to feed millions and millions of hungry mouths because of not keeping pace both food production and the population explosion. According to the FAO of United Nations, every day arable land is shrinking because of urbanization, because of industrialization, and because of various anthropogenic activities. That means there is no scope to increase the arable land. So whatever shrinking land, because of various anthropogenic activities, we are losing the land, that too particularly important arable land. Whatever the available land, we need to grow and produce more. That should be our motto. And FAO, Food and Agriculture Organization of United Nations Organization, estimates on an average, global average, only 2 out of every 5 people are having balanced 2 meals per day. So this is the FAO estimate. I'm telling you, Food and Agriculture Organization of United Nations Organization estimated that on a global average, irrespective of the status of the countries, on the global average, only 2 out of every 5 people will be having balanced 2 meals, nutritionally balanced 2 meals per day. Remaining 3 people out of 5 on a global average going to bed either half-filled or malnourished or completely empty. So this is the status of food production and the supply. Although we produce more, but affordability of food is question mark. Okay. So what is the alternative? And in addition to this, this is one part of the story. The other part of the story is whatever we produce, the agriculture produce, almost, this is again the estimate of FAO, of United Nations Organization, almost 30 to 35% of the total agriculture produce is going as waste towards pests and diseases. That means the pests and diseases will take away their contribution of 30 to 35% of the total agricultural produce. Total agricultural produce includes cereals, oil crops, vegetables, pulses, all these things, fruits, all these things. About 30 to 35% of the total agricultural produce is going as waste towards pests and diseases. So among diseases, so we know different groups of microorganisms which are responsible for causing the disease. That is true in case of plants, that is true in case of animals, and that is true in case of even human beings. Okay, so fungi is the dominating group among the disease causing organisms, either in case of plants or in humans or in animals. Fungal microorganisms will take away about 15% of the total agricultural produce. Next comes bacterial microorganisms. The groups of bacterial microorganisms will take away about 10 to 12% of the total agricultural produce. Remaining agricultural produce out of 35% is share taken by either viruses or insects and other pests. So these groups of microorganisms, either fungi or bacteria or insects or nematodes or viruses, will take away their share, and that will amounts to the total amount is about 35% of the agricultural produce. So if you can save that amount of agricultural produce from the pests and diseases, you can think of providing that saved agricultural produce to the millions and millions of hungry mouths. Okay, although microorganisms are omnipresent. So microorganisms are present everywhere. They are present on the air, they are present in the soil, they are present in the water, they are present inside the human being. And according to one estimate, the gastrointestinal tract of human being will be having almost 600,000 different kinds of microorganisms. And the medical report further says, your digestive system will function normally only when you have proportionate concentration of all these microorganisms. Irrespective of your food habit, irrespective of your drinking habit, all these microorganisms which are contributing significantly for the proper digestive system functioning. Okay, so these microorganisms are omnipresent. Most of them are beneficial. Only 10% of the microorganisms are known to cause diseases in case of either plants or animals or human beings. Remaining 90% of the microorganisms are beneficial to human beings. So microbes are omnipresent, they are present everywhere, they are present inside the human body, they are present outside. You name any of the conditions, even at very high temperature of 100 degrees centigrade, you have a presence of microorganisms. Even at very low temperature, you have the presence of microorganisms. So you have some of the microorganisms invariably work and reproduce only in complete absence of oxygen. That is the scenario of the microorganisms. But we are concentrating only on 10% of the microorganisms which are harmful to human beings, which are known to cause diseases in case of plants, which are known to cause diseases in case of animals, which are known to cause diseases in case of human beings. So only those 10% of the microorganisms which will take away about 35% of the agricultural produce. So with this background information, to manage any of the diseases, either plant disease or animal disease or human disease or foodborne disease, the first and foremost important thing is to diagnose those diseases properly. By any chance, if you wrongly diagnose those diseases and recommend the management strategy, which will all going as waste. So if you want to manage the diseases properly, disease diagnosis is playing a pivotal role. If you identify the disease properly, if you diagnose the disease properly, it is easy for any one of us to propose any of the management strategies. Management strategies include either chemical or physical or biological or any of these, biotechnological, any of these management strategy you can recommend when you only identify the disease causing organisms properly. That means when you diagnose the disease properly, it will be easy for any one of us to propose the management strategy for that particular disease, control strategy of that particular disease. So if you wrongly diagnose the disease, whatever control strategy you recommend, that is going as waste, in addition to the season, in addition to all other agricultural inputs. So disease diagnosis is playing a pivotal role in the management of the disease. So if you manage the disease, you can save some amount of the agricultural produce. Otherwise, there is no other alternative go. As I said, there is no scope to increase the agriculture land, because arable land, we are using it for various anthropogenic activities, either in need or not in need. So totally we are losing day by day the agriculture land. That means the production area itself is reduced. And whatever in the reduced area we are going to produce, that amount of produce, about 30-35% of the agriculture produce, is going as waste towards pests and diseases. And as I said, if you want to control the disease, if you want to manage the disease, it is your primary duty. The first and foremost prerequisite is to properly diagnose the disease. And you have various methods to diagnosis. Basically, if you take to identify a disease causing organism, as we all of us already know that, you need to isolate the organism and subject that particular organism to the cause postulates. You need to isolate, purify, then you need to isolate the organism from the disease causing or symptomatic portions of the host. Here, host includes either plants, animals, or human beings. So whatever the symptoms which are showing, that is manifestation of the disease, from that symptomatic host, you need to isolate. And while isolating, as I said, microbes are omnipresent, they are present everywhere. You may have innumerable number of microorganisms. So you need to purify those microorganisms which are responsible for causing these particular symptoms. Once if you purify, then you need to, that means you need to culture the organisms away from the host in the laboratory conditions. Once if you culture, then if you re-inoculate those organisms to the host, and that host should produce similar symptoms. So these are the Koch postulates. Robert Koch is a medical practitioner proposed to identify the disease causing organism in any of the hosts, either plants or animals or human beings. So you need to prove the Koch postulates. And you need to grow the plants. In case of plants, you need to grow and observe carefully the symptoms of that particular plants. And you need to subject those isolated pathogen to the Koch postulates. And then you can attribute, okay, this particular symptom is due to this particular disease causing organism. Once if you know the disease causing organism, then you can say, okay, this is the causal organism and this chemical you need to use for the control of this organism. Or this biological method we need to use. Or this biotechnological method you need to use to control this particular organisms. Okay. So in that identification, as I said, our laboratory in the Department of Biotechnology, University of Mysore, since the last 1 decade almost, working very hard to get some of the molecular diagnostics, because this is the recent trend, use of DNA to identify the disease causing organism. Use of nucleic acid to identify the disease causing organism. Because of late, one of the fascinating trend in the identification strategy is to use the DNA in this particular microorganism. So, as I said, the agricultural scenario of India. India is an agricultural country. I need not have to emphasize all these things. And bacteria constitutes the major risk factors in disease causing organisms. And the second, next to fungi, the second group of organisms are bacteria. And India is considered as fruits and vegetable basket. We are producing enormous amount of fruits and vegetables. We are second in the production, second to the China. First being the China, and we are the second in producing the fruits and vegetables. And these particular groups of microorganisms, which are the bacteria, unlike in any other groups of microorganisms, like fungi, where fungal microorganisms can enter the host by piercing the cell wall. whereas bacterial microorganisms cannot do so. Bacterial microorganisms always finds the natural openings like stomata or hydrothodes or nectarithodes or wounds, manual injuries to get entry into the system. And these bacterial microorganisms reproduce or produce enormous number in the intercellular spaces and reaches throughout the plant body that will be systemic in nature. So lack of effective control measures makes the disease management very cumbersome and economically not feasible. As I said, if you identify properly the disease causing organism, then effective management strategy you can recommend. If the identification is wrong, whatever recommendations you propose, all those things are going as waste. And I said, in our research laboratory, our students are using plants, seeds as waste material. In addition to plants and the seeds, we also use food because foodborne pathogens also posing major threat. And some of the domestic animal pathogens like cows, where we use dairy products, etc. So what is food? Food is a complex substance derived from either plants or animals, which we use mainly for nutritional purpose. And the food safety is monitored routinely by agencies like FAO, IFIC, FSSAI, etc. This is Indian and the others are American and European. And food contaminated with number of pathogens. Those pathogens can transmit by various routes and cause major health issues, which lead to the life-threatening human diseases. So number of foodborne pathogens are reported by World Health Organization and CDC, Committee for Disease Control, Campylobacter jejuni, Listeria, Salmonella, Shigella, and the others. Staphylococcus aureus, which are most frequent cause of food poisoning. So food poisoning is caused by whether Shigella flexneri, Salmonella typhimurium, Staphylococcus aureus, Listeria, and Campylobacter. These are the known foodborne pathogens which cause food poisoning in case of human beings. And particularly Staphylococcus aureus, Staph aureus or S. aureus, colonized approximately 30% of the human population and resides predominantly in the nares, anterior nares, harmlessly on the skin and in the GI tract, gastrointestinal tract. This particular organism, that is Staph aureus, causes skin and soft tissue infections, SSTIs, aggressively into bacteremia and infective endocarditis or sepsis. Sepsis is also caused by Staph aureus. I was telling you the conventional identification techniques by isolating the pathogen and prove the Koch postulates. Identification through conventional methods are being replaced by nucleic acid based detection techniques, NAT based tests, because there are no drawbacks or demerits of those conventional or traditional methods. Sometimes those may be complex to perform. And identification of these organisms with a bulk sample, enormous amount of sample, can be subjected to in a very short span of time. So you have got a great responsibility to identify the disease causing organism or to test various samples within the short span of time. That is still a challenging task to the scientific world. Developing easy and sensitive method for identification of pathogens can give more attention about the food safety before they are released into the market for consumption purpose. This is a sort of review which our students have published in the important journal Review of Critical Reviews in Food Science and Nutrition. Then actually diagnosis starts from here. The traditional methods or the conventional methods is to isolate the pathogen, purify, culture in the laboratory condition that is away from the host, and again the pure form of the pathogen should be re-inoculated to the host, and host should produce similar symptoms. That is the postulates. The importance of diagnosis. If you identify that, if you diagnose the disease properly, then management strategy will be easy. And traditionally, cultural methods are very, very slow. And of late, the PCR based, the amplification of nucleic acids are promising methods for the rapid identification of low numbers even. But the drawback of these PCR based methods is, even if the whole the pathogen is dead, because its nucleic acid will be there, the DNA will be there, it will give us the positive result. That is false positive. What are the host and pathosystems which we have used in the present studies? We have used the following host pathosystems. The first four are mostly from the plants, vegetables, like Xanthomonas xenopodis petavar vesicatoria, which is a bacterial spot causing bacteria in case of chili, brinjal, and the tomato. Xanthomonas oryzae petavar oryzae, which is bacterial leaf blight causing pathogen in case of fries. And Xanthomonas campestris petavar campestris, which is the block rot causing bacteria in case of cold vegetables, that is cabbage, broccoli, nolla, etc. And one of the important deadly pathogen is Ralstonia solanacearum, which is the bacterial wilt causing bacteria in case of chili, tomato, brinjal. This particular pathogen, Ralstonia solanacearum, can cause in more than 500 plant species, which belongs to 50 different botanical families. That means which has got very, very, very wide host range. Mostly the host and pathogens are very, very specific, but this is exception. Ralstonia solanacearum can able to initiate the disease in more than 500 different plant species, which belongs to, which spread across more than 50 botanical families. And in addition to this, we have also used in the present study Staph aureus, Staphylococcus aureus, which is important foodborne bacterial pathogen. And one of the cattle disease, that is leptospirosis, leptospira, which is a zoonotic disease. Zoonosis is nothing but the transmission of disease from the domesticated animals to human beings and vice versa. Zoonosis. So these are typically the symptoms of tomato plants, which are either spot infected or wilt infected or blight infected. So tomato plant showing typical symptoms of bacterial spot, you can see. And upon the advancement of the disease, you can notice complete death of the pathogen. On the fruits, these are showing typical symptoms of bacterial spot, so that reducing the marketability of the fruits. Though the consumption of these spot bacterial infected fruits is not causing any of the harmfulness to the human beings. There are no reports. That's why, but still marketability of this particular fruits is reduced. This is block rot disease symptoms on the cabbage plants. I told you bacteria mostly enters into the host tissue through stomata or idototes or nectarithodes and reproduce in the intercellular spaces enormously and cause necrosis. You can see the V-shaped necrosis on the cabbage plant. Similarly, bacterial leaf blight of rice caused by Xanthomonas oryzae petavar oryzae. From the distal end of the leaf, it starts towards the proximal end of the leaf. And this is how, based on this, you can say how much is the infection rate of rice plants because of bacterial leaf blight bacteria, that is Xanthomonas oryzae petavar oryzae. So we have collected the seed samples from the various sources, and we have subjected all the collected samples from various sources to the traditional or conventional methods, like which are mostly International Seed Testing Association recommended, direct plating or liquid assay. And we need to isolate and characterize the disease causing organisms by subjecting those bacteria, pathogenic bacteria, to the morphological characterization, biochemical characterization, or physiological characterization, and the molecular characterization of these bacteria using universal primers, 16S or RNA primers, because those are universally present in almost all prokaryotic organisms, sufficiently conserved and of adequate length. This is the reason why we are using 16S or RNA. This is the morphological characterization, colony morphology, which is showing from the semi-selective media growth, either universal medium or semi-selective, sometimes the universal. This is Xanthomonas campestris, Pathogen campestris, Ralstonia solanacearum, which is bacterial wilt causing bacteria, Xanthomonas oryzae, Pathogen oryzae, bacterial leaf blight causing bacteria in case of rice, and Xanthomonas xenopodis, Pathogen vesicatoria, which is bacterial spot causing bacteria in case of chili, tomato, and the brinjal. So actually, from this point onwards, we are going to address or discuss what are the different PCR techniques which can be used to isolate and characterize the pathogenic microorganisms. Only characterization, not isolate. So one of the important modifications in the PCR, polymerase chain reaction, single strand conformation polymorphism. This is the slight modification of PCR, single strand conformation polymorphism, which is most widely used technique for the identification of point mutations in case of human beings. So this technique, which we are going to exploit it for identification of bacterial microorganisms. The 16S rRNA gene, whose use is considered as a gold standard for prokaryotic identification. For bacterial identification, we always use 16S rRNA gene because these are universally present. These are sufficiently conserved and these are of adequate length. So this SSCP, single strand conformation polymorphism analysis, is being done after the amplification of PCR fragments. So SSCP analysis of PCR amplified fragments of 16S RNA gene has more recently been used as an alternative to genomic sequencing for the proper identification of the prokaryotic microorganisms. So the mechanism of this SSCP, single strand conformation polymorphism, is just, so once if you subject the DNA for PCR amplification, it will amplify. And after the amplification, the amplified product should be given either heat shock or cold shock. If you give heat shock or cold shock, the single stranded DNA will fold into different conformations. This is different conformational changes. And remember, these changes of conformation in the single stranded DNA is always specific to a given individual subspecies, not species, even at the level of subspecies. So after the amplification of PCR, the target sequence, the amplified product is denatured to 2 single stranded DNAs, ssDNAs, and subjected to non-denaturing phase, that is polyacrylamide gel electrophoresis. Under this non-denaturing conditions, single stranded DNA has a secondary structure. These are the secondary conformation. Secondary structure that is determined by the nucleotide sequence. The mobility of this single stranded DNA depends on the secondary structure of the amplified product. And if you run the gel, it will give us the unique banding pattern or unique fingerprinting. This unique fingerprinting is nothing but, though it is universal to all microorganisms, but the banding pattern is always specific to a particular subspecies or a particular pathogen, not only the genus and the species, even at the subspecies level, this is specific. So PCR single stranded conformation polymorphism, the procedural details includes isolation of bacterial DNA and amplification using PCR universal primer. And once if you give heat shock and cold shock, the fingerprints will be obtained. PCR SSCP fingerprints will be compared with the other phytobacterial pathogens for the proper validation of these fingerprints. So this is how the fingerprints of the various organisms, particularly Ralstonia solanacearum. All these are different isolates of Ralstonia solanacearum, and Ralstonia isolates are collected and confirmed by various physiological characterization tests. and which is unique binding pattern you will get, because if it is Ralston serum, then only it will use us the unique fingerprinting. Otherwise, it is not unique. So this is the unique fingerprinting pattern of Xanthomonas xenopodis Pathogen Caturia. When you compare these two, when you Xanthomonas varese, Pathogen varese, when you compare all these three, it is like this. Individually, at one particular base pair, Ralstonia solanacearum, Xanthomonas varese, Pathogen vesicatoria, Xanthomonas varese, Pathogen varese, and the other non-pathogenic organisms like E. coli and the negative control and molecular weight marker. So what are the applications of this PCR SSCP? SSCP has the simplicity in the usage without any special equipment, and radioactive labeling is also not necessary. Although DNA sequence has become more common technique nowadays, but the equipment is still, sequencing equipment is not available in most of the laboratories. That is why this SSCP can solve the problem of those sequencing technologies. Then you need not have to isolate the DNA. Directly you use pure colonies, that is colony PCR SSCP. In the normal PCR SSCP, we need to isolate the DNA and then subject it for amplification. But here, you need not have to isolate the DNA. Directly colony itself can be used. Colony PCR requires isolation of pathogen, but does not require isolation of DNA, subculturing and isolation of DNA or any other pre-treatments. So colony PCR has an advantage of its rapidity and simplicity. So you need to isolate the DNA, you need to isolate the culture, not DNA, purify pure bacterial culture and use 6' RNA primers, and also species specific primers can be used to amplify this SSCP. Colony PCR SSCP fingerprints will be compared with other phytobacterial pathogens for validation. This is the DNA amplification, and colony PCR SSCP fingerprints, where we have not subjected the bacterial strains for isolation of DNA. Directly, we have used the bacterial colony itself. Colony PCR SSCP fingerprints of Xanthomonas xenopodis Pathogen vesicatoria and Xanthomonas Ralstonia solanacearum and Xanthomonas xenopodis Pathogen vesicatoria, along with the positive and the negative controls. So application of this colony PCR, as I said, you need not have to isolate the DNA. Directly you can use the colonies, but only thing is you need to have the pure form of colony. But even if you identify, even if you want to identify the one particular colony with a mixture of different bacteria, then you need to use that particular species specific primer. If you use species specific primer, it will amplify only one particular species of the organism, and it will not amplify any other, though you might have used the mixture of bacteria. So then multiplexing PCR, you need not have to use one colony. You can use many different organisms. Sometimes we have used some fungi and some bacteria. Multiplexing PCR or multiplex PCR, either duplex PCR or triplex PCR or multiplex PCR. But only thing is here, the difficult part is we need to standardize the proper TM melting temperature and also PCR conditions. Multiplex PCR is a widespread molecular biology technique for the amplification of multiple targets in the same PCR equipment. You can just imagine ingredients of the PCR will be same with a lesser quantity, but you can amplify many different colonies, though they belongs to fungi or bacteria. Provided you need to properly standardize TM melting temperature and the PCR conditions. More than one target sequence can be amplified using multiple primer pairs in a same reaction mixture. Thereby you can reduce so much of ingredients because PCR ingredients are costly. Our present research exploits this method for pathogen identification. For example, Xanthomonas campestris, which causes black rot of cabbage, and also Ralstonia solanacearum, which causes bacterial wilt in case of tomato. That means the groups of the mixture of tomato pathogen and cabbage pathogen can be identified using same PCR mix or PCR ingredients. And these are the genes which you have used, HRP genes. The HRP genes encode type 3 secretion protein and the multiplex PCR fingerprints. Normally we obtain at 619 base pair for HRPF gene from Xanthomonas campestris Pathor campestris, and the 360 base pair section of ITS region from the Brassica species. So here in this PCR, we have identified both pathogen and the host in the same PCR mix, and in the subsequent PCRs, we have used both the host and both the pathogens. This is the procedural details, how exactly you can take up multiplexing PCR technique, the garostil electrophoresis, and isolation of plant pathogen characterized traditionally or conventionally, then subject it for traditional identification, then you subject it for nucleic acid based amplification. So this is the PCR amplification of Xanthomonas Pathor, and with a species specific primers, which compared to 100 base pair ladder in the molecular weight mark. And this is PCR amplification of Ralston's serum. And when you run these two separately, you will get 331 base pair and 281 base pair. But when you run in the same duplex PCR, this is how you will get both 331 base pair and 281 base pair in the same gel. That means you can use multiplexing PCR for the identification of 2 or 3 pathogens along with the host in the same PCR reaction. This is the application. Multiplex PCR method plays a crucial role in the identification of several pathogens in a same single reaction mixture, so that you can reduce ingredients of PCR. The procedure developed from the present studies overcomes the laborious or conventional techniques or the characterization of phytobacterial pathogen. These results are published in this particular journal. So seed samples can also be directly subjected to multiplexing. You can subject the seed samples, you can subject the food, or you can subject any of the unknown pathogens. And you will get the amplification provided if you use species-specific primers. And this technique developed in the present study is useful for international sanitary surveillance of seed exchanges. These results were published in all these research journals. Development of diagnostics for leptospiral identification, that is zoonotic disease. Zoonosis, as I said, transmission of the disease from the domestic animals to human beings and vice versa. Either human beings to domestic animals or domestic animals to human beings. In this leptospiral identification, what we have done, we have isolated the DNA, we have designed the primers, and we have standardized the PCR. And we have also carried out some of the other steps like transformation, isolation of plasmid, and finally PCR SSCP. That means same PCR SSCP which we have used for identification of seed-borne pathogens, domestic animal-borne pathogens, or even food-borne pathogens. These are the primer pairs which we have used for identification of leptospira, which we have only designed. These are the thermal cycling conditions which we have used for identification of zoonotic disease. So PCR products of leptospira isolates when amplified with this particular primer, LipL, lipoprotein L32 primer, and RE digestion, restriction enzyme digestion, with the same LipL32 gene of leptospira identification. This is the PCR products of leptospira along with LipL41, that is LipL32, this is LipL41 with the same RE digestion. The procedural details of this: preparation of ligation mixture, the transformation. Ligation mixture was added to ice cold 200 microliter of competent cells, incubated, and heat shock was given, and you will get the competent cells, blue white screening we call. This is the insert release of LipL41 and LipL32 in case of leptospiral identification. After this, we have subjected same amplified product to SSCP analysis. The PCR amplified products were first evaluated for purity, and we have subjected for single strand conformation analysis, and we obtained this particular gel. That means PCR SSCP fingerprinting patterns of Leptospira species. So this was visualized on non-denaturing. This is Leptospira hictioemeraceae, different species of Leptospira. Leptospira japonica, Leptospira astigilis, Leptospira pyrogenes, like that, along with the marker and the negative control. And this is the maximum likelihood tree, phylogenetic analysis, construction of phylogenetic tree, and all this Leptospira belongs to this particular cladogram, and E. coli is different pathogen, that is non-pathogenic organisms. Then comes foodborne pathogens, Staphylococcus aureus. Mostly we have isolated the Staphylococcus aureus from various vegetarian, non-vegetarian, dairy, bakery, and processed products. And we have representative isolates we have submitted to NCBI gene bank. And this is moldy biotyper, which is one of the important diagnostic tool. Mysore matrix assisted laser desorption ionization is the moldy. Moldy biotyper is unique experiment which we have developed for isolation and characterization of Staphylococcus aureus, which is one important foodborne pathogen. So a collection of different food samples, isolation of Staph aureus, and isolation characterization through this moldy biotyper, either by direct smear or through protein extraction. And the one group can be subjected to biochemical characterization, which is the traditional method, conventional method, and the molecular approach after isolation of DNA and amplification through PCR using universal primers and the species specific primers, and again subjected to PCR single strand conformation polymorphism. And when you use species specific primers, and you need to subject them for sequence analysis, and you can submit those sequences to NCBI data bank. These are the different food samples which we have used for isolation of different foodborne pathogens: Staphylococcus aureus, Bacillus cereus, Typhimurium, E. coli, Shigella, etc. These are the colony morphological characters, different pathogens on the species specific chromogenic medium: Staphylococcus aureus, Shigella flexneri, Salmonella typhimurium, Bacillus, E. coli, Vibrio, etc. And these staphylococcal species associate with the different food items. How many vegetarian items are showing Staphylococcus aureus, Staphylococcus epidermis, and Staphylococcus hemolyticus? These are the three different species of Staphylococcus: vegetarian, non-vegetarian, dairy products, and the other food sources. Foodborne Staphylococcus aureus, we have concentrated only on Staph aureus. So although other important foodborne pathogens are there, but we could able to concentrate only one staphylococcal species by subjecting that into various biochemical physiological characterization methods. And these are the accession numbers after submission of gene sequences to NCBI gene bank. This moldy biotyper, which is used in the identification of the Staphylococcus aureus, you take the sample culture either in the solid medium or in the liquid medium matrix. You can load the sample on this particular plate, and it will be provided to a machine to read, and it will identify, and it will give us the spectra because of the cell wall material which constitutes this particular chemical compound, which will give us the spectrum. So this moldy biotyper is playing an important role or crucial role in the identification of the different disease causing organisms. So how exactly it will give us the identification? It is based on the color and also the symbols, which is this range of 2.3 to 3, highly probable species identification with the symbols of +++. And 2 to 2.29, secure genus identification and probable species identification, only 2+. And 1.7 to 1.9, probable genus identification and not species identification. And others is not reliable identification. This is how the molded of MS spectra of Staphylococcus aureus. Different foodborne pathogens on molded of spectra other than staph, that is E. coli, Bacillus cereus, etc. The specific identification of Staphylococcus aureus as assessed by species-specific primers. When we use the species-specific primer like thermonuclease gene, NUC gene we call, it will give us the proper identification, because all this data we have compared. We have subjected those Staphylococcus aureus for traditional identification, conventional identification, biochemical characterization, and molecular characterization. And we have compared the entire data into for the secure or the confirmation of this identification. The PCR SSCP binding patterns of mostly other foodborne pathogens, which gives the SSCP is working well even for identification of foodborne pathogens. So PCR SSCP is working well not only in the identification of seed-borne pathogens, not only the identification of leptospira, that is animal-borne pathogen, and also foodborne pathogens. So we have successfully characterized the foodborne 160 methicillin-resistant Staphylococcus aureus (MRSA) strains in the present study involving 50 different food samples and showed represented moldy biotyper as an alternative, robust and sensitive tool for the secure identification of Staph aureus. So in addition to the Staph aureus moldy biotyper analysis, we have also developed a proper PCR SSCP fingerprints for the identification of foodborne Staphylococcus aureus. That means we have compared both these data. We have compared the moldy spectra along with SSCP fingerprints. So both of them exactly confirms, okay, whatever you have used the microorganism, you have used bacterial pathogen for identification is Staphylococcus aureus. So this, along with, in addition to these two, we have also subjected that Staph aureus for colony morphological identification, biochemical identification, and growing on its species-specific chromogenic medium. All these identification results are exactly concurrent, so that whatever technique we have used, either it is moldy biotyper, or it is PCR SSCP, or colony PCR, or multiplex PCR, all these things are proving very well, or working very well in the identification of this disease-causing organism. So this entire data we have published in this particular scientific journal, and immediately that particular boy who has worked for this particular work has got an offer from USA to postdoc. Then in addition to these things, we have also carried out some of the other loop-mediated isothermal amplification with the help of single species-specific gene targets. We have designed the primer for SPA target gene, and we have standardized loop-mediated isothermal amplification, which is again the sensitive and the robust. And immediately you can subject any number of samples for the identification of those organisms with the help of loop-mediated isothermal amplification. In this particular technique, you can not only run the gel and get the bands, you can also see in the visual change in the color, which will be showing. So these are the primer primes which we have used for LAMP primers we call. LAMP assay is loop-mediated isothermal amplification assay. And this is how the thermal cycling conditions, the isolated Staphylococcus aureus confirmed by LAF F3 and B3 primers. So these are the different strains which are used for LAMP reaction. Bacterial strains used in the assessment of the specificity of loop-mediated isothermal amplification. Specificity of the LAMP assay. These are the binding patterns of one particular isolate, the other isolate, and the other isolate along with the positive control, along with the molecular weight marker. And you can see the change in the color in the append of tube, so that you can directly use this particular technique. You need not have to do this particular running the gel and the others. The confirmation of LAMP products by HNB, that is stain on 2% agarose gel. Sensitivity of conventional PCR and in-tube LAMP reactions. So we have compared both the data, in-tube LAMP reaction and the LAMP PCR amplification. So sensitivity of conventional PCR assay along with in-tube LAMP reactions, which are exactly in concurrence with, along with the 10-fold serial dilution of Staph aureus with one particular University of Mysore 090 genomic DNA we have used. So we have developed a versatile loop-mediated isothermal amplification LAMP assay for identification of multi-drug resistant Staphylococcus aureus. And we have published all these data in the various scientific journals like medicinal Chemical Communications, Medicinal Chemical Communications, Food Research International, and Arabian Journal of Chemistry recently. Actually, it is 2021, not 2020, because 2021 January issue we got this paper published. I think this is the end of my lecture. If you have any questions, you can ask me. Thank you.
Thank you, sir.
Thank you.
Thank you, sir.
Question.
Hello.
Yes, sir.
Hello, sir. That MALDI instrument, sir, how
Yeah.
it identify the multiple of organisms? Sir?
Actually, along with the MALDI instrument, they provide the pre-coated plates, just like ELISA plates.
Yes, sir.
ELISA plate, pre-coated chemical components will be there, which are specific to cell wall chemicals of one particular bacteria, one particular fungi. And in the entire South India, we have only one instrument in Coimbatore.
Yes, sir. Yes, sir. Thank you, sir.
Yes, sir.
Excuse me, sir.
Yes, sir.
Sir, I'm Dr. Sarath. I'm working as an assistant professor in Dr. SRK College. Sir, during your lectures, you have quite often mentioned the specific primer.
Yes.
How did you design this specific primer?
This is very good question, actually, sir. For in all microorganisms, you have various genes, and there are softwares nowadays. You can use Primer 3 software, and you can feed whatever gene you want to design the primer. For example, if I want to design a primer for one particular gene in one particular bacteria, one particular gene, for example, HRP gene, hypersensitivity gene, you can feed that hypersensitivity gene in that Primer 3 software. It will give you species specific primer. And this is not only software, there are other methods also where you can design the primer. Totally, that particular primer, if you use whatever the amplification product you expect, that should be amplified. That is how you need to design the primer. And of course, there are softwares which will give you the exact picture of this particular primer. Say if you design the primer for one particular color, for example, one particular taste, for example, there are genes which are responsible for color. There are genes which are responsible for taste. There are genes which are responsible for infectivity. There are genes which are responsible for disease causing ability. So all these genes which you need to study enormously in the literature, and then you need to feed that particular gene in the software. It will give you the primer which are species specific primer. That means if you use that primer for the PCR amplification, it will correctly amplify. It will amplify only one particular organism. It will not amplify in any of the other organisms. That is how you need to design the primers.
But sir,
Yeah.
here I have this doubt that microbial genomes are quite dynamic. And there
Exactly.
are also cases of lateral gene transfer in the environment.
Exactly.
So how much you are sure that the specific gene you are amplifying is specifically comes from that particular bacteria?
Exactly. That is the reason for some microorganisms we have designed 2 pairs of primer which are species specific. For some microorganisms we have designed 3 pairs of primers. For some we have designed 4 pairs of primers. So when you use all these 4 pairs of primer, it should amplify only one particular xanthomonas, for example. It will not amplify in any other either Pseudomonas or E. coli. So even if you mix the culture and try to amplify, whatever your designed primer, it should amplify only xanthomonas, not the Pseudomonas and the E. coli. So that is how you need to design. Of course, this designing of primer is always in a parallel with the conventional methods of identification. The traditional methods of identification, Koch postulates you should never forget. You need to isolate the pathogen from the symptoms, and you need to culture away from the host, and you need to purify. Then you need to reinoculate the susceptible host. Then you should be in a position to get the similar symptoms. then only whatever isolate organism which you are attributing is either Xanthomonas or Pseudomonas or E. coli or Erwinia or Clavibacter, any of these things like Fusarium, Aspergillus, Druxlera, any of these fungal microorganisms. So you need to go both hand in hand, not only either conventional methods or molecular methods. So both conventional methods, for example, say by using conventional methods, it may take at least 1 week's time to grow the plant, to get the symptoms, then isolate the symptomatic portion, then isolate the pathogen from the symptomatic portion, then characterize the pathogen, then you subject the pathogen to the Koch postulates, then re-inoculate to the host and get the symptoms. This will take until 1 month. Okay, this is proved. The same isolate the pathogen from the same organism or the host, subject it for various molecular methods. If it gives you the correct results, then you use, otherwise don't use. That is how all these molecular methods are just an assistants to conventional methods. So conventional method is always king. Proving Koch postulates is always king. You cannot do only molecular methods. Conventional methods, in addition to conventional methods, as a supportive data, you can use molecular methods. But over a period of time, when you prove, for example, Xanthomonas you have proved, then subsequently in the next time onwards, you need not have to raise the plant in the field. Directly you submit to the laboratory conditions. Directly you amplify and it will give you the correct results. That is how you need to do. You cannot simply depend only on molecular methods or only on traditional methods. Both the methods you should club together and work for identification.
Okay, sir. Thank you, sir.
Thank you.
Sir, I am Somlingam from Gadag, sir.
Yes, sir.
Sir, whatever the methods that you have developed for the detection of the bacterial detection in the food
Yes, sir.
and the food plants, these methods are also used for the identification of the viruses or molecular diagnostics of the viruses, sir.
You can standardize these techniques for the detection of viruses also, sir, but we have not standardized in our lab. But there is a scope to standardize. Maybe here and there you need to reduce the temperature, increase the concentration. All these minor changes, minor adjustments you need to do. But you
Yes,
can
sir.
subject any of these viruses to same molecular methods.
Yes, sir. Yes, sir. Yes, sir.
Yes, sir. Thank you.
Thank you, sir.
Yes, sir.
Excuse me, sir.
Yes.
Hello, sir. My name is Dr. Nazir Basha, sir.
Hello.
Yes, sir.
Hello.
Basha, sir. From where are you?
Yeah, I am from C. Abdul Hakim College, Tamil Nadu, sir.
Yes, sir.
Sir, which is the best DNA extraction method, sir? That means snap cell method or chemical extraction method?
Sir, for these extraction methods, you cannot say this is method best, this is not best like that.
Which is efficacy, sir?
You need to standardize, sir. It depends on the individual organism. For some organism, the other method may be good. For some other organism, this method may be good. You need to standardize and see for given microorganism, which is the best method. For our Xanthomonas vesicatoria or National, whatever we have used is best method. It is not universal to all, sir. It depends on the individual microorganism. That means you need to standardize the particular method for the isolation of DNA from either Xanthomonas or Pseudomonas or Staphylococcus or Leptospirosis.
Okay, okay, sir. Thank you.
Yes, sir. Yes, sir.
Sir, I have one question. I am Niranjan.
Yes, sir. Niranjan, tell me.
Sir, for all this base for the identification of Xanthomonas at the species level,
Yes, sir.
and suppose in a strain level,
Yeah.
the postmost is the isolation of a DNA followed by a identification by 16S ribosomal RN.
Yes, sir.
So in that one, after sequencing, we have designed the primer for the species level identification.
Yes, sir.
Then followed by there will be a strain variant.
Correct.
For that we have isolated these samples. Further you have grown your culture. Then all these repetition only you are capable of identification at the species as a strain level.
Yes, sir.
Okay. Thank you, sir.
Yes, sir.
Hello.
Yes.
So, sir, actually, the false positive in the RT-PCR for corona,
Yes,
whether
sir.
the rate is too high or
It is not too high, sir. It is actually reports say it is less than 5%. If you take less than 5%, I feel it is too high. It should be less than 1%.
Okay. Thank you, sir.
Where are you, sir? Sir, from where are you? Babu,
Sir, I'm from Trichy, sir.
Kerala.
No, Tiruchirappalli.
Okay, sir. Trichy. Okay. Tamil Nadu.
Thank you, sir.
Yes, sir.
If no more questions, dear participant, you have any questions, madam?
No, sir. No, sir.
Can I come to the vote of thanks?
Niranjan, sir.
Yes, sir.
Now the madam has put in the chat box. She wants to propose the vote of thanks.
Okay, thank you.
Good evening, sir.
Good evening, madam.
I am Dr. Devika from Sharda Vilas College of Mysore. I take the privilege of proposing vote of thanks to Professor Umesha, Senior Professor from Department of Studies in Biotechnology. Sir, you have delivered a wonderful talk on molecular diagnostics of detection of pathogenic bacteria. Maybe it was possible for you due to the vast experience and your extensive work on that specific field, sir. I would like to thank on behalf of all the participants and on behalf of my own self and on behalf of HRDC Mysore. But sir, at the same time, I have a request from my side and I hope all the participants also agree with this. I don't know exactly whether they like it or not. When we attend such refresher courses, especially when it comes to topics like this, molecular diagnostics and all, we want to have some experience, practical experience. Of course, now it is not possible. But my sincere request is from your department, why don't you organize a hands-on workshop, something like that for undergraduate teachers like us, so that we can have hands-on experience regarding this.
Madam, actually, we have proposed one hands-on workshop to DST. It has approved. It has approved. Soon we will announce.
Thank you so much, sir.
But madam, every year we are planning to conduct because of lack of funds. You know, all these things require majorly the fund requirement is important.
Yes.
Since the last 5 years, we have been applying funding for DST.
Okay.
Only this time we got the approval. Soon we will be organizing.
Okay. Thank you so much, sir.
Thank
Please
you, madam.
wait, sir.
Thank you.
Thank you, sir. Thank you very much.
Thank you so much, sir. Chandrasekhar.
Thank you, sir.
Spelling ತಪ್ಪಾಗಿದೆ ನಿಮ್ದು ನೋಡ್ಕೊಳ್ರಿ. Thank you, Chandrasekhar.
Thank you, sir.
Thank you.
Thank you so much, sir.
Yes, sir.
Thank you so much.
Yeah.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you. So shall I sign out?
Yes, sir.
Yes, sir. You can.
Yes, sir.
Okay.
Okay.